Hey Beaks, today we're going to dive in even deeper into adaptive or acquired immunity. All right, so here we go. So the adaptive immune system has two major branches, right? And it's a protection against infection of body fluids and body cells. In the humoral immune response, we make antibodies, right? We help to neutralize or eliminate toxins and pathogens in the blood and lymph. So we'll talk more about that. And in the cell-mediated immune response, we make these specialized killer T cells that destroy affected host cells. Also is a good combatant of cancer. Now we have an all-star, in my opinion, of the immune system, and that is a helper T cell. Okay, and this type of T cell, or helper T, triggers both humoral and cell-mediated immune responses. Signals from helper T cells initiate production of antibodies, so this is part of the humoral response, right? And they neutralize pathogens, and they also activate T cells. Okay, and this kills infected cells, or like I said, sometimes cancer cells. Now, to start this process, a lot of times what we need is an antigen-presenting cell. Okay, and these guys are going to have class 1 and class 2 MHC molecules on their surfaces, which we'll discuss in just a second. Class 2 MHC molecules are the basis upon which antigen-presenting cells are recognized. So basically, these major histocompatibility complex, these molecules, they're self-proteins. Okay, because if you remember what an antigen is the little part of the pathogen, okay, that your immune system is going to recognize. Actually, remember, it has like a little epitope on it that the antigen will fit inside of an antigen receptor, and that epitope will bond very snugly, and boom, there you know you have the right molecule. Well, you have to know, am I seeing an antigen from a good guy that's being presented to me, or from a bad guy because it's part of the bad guy's cell, like a virus or a bacterium? That's the whole beauty of MHC2, okay, or MHC1. MHC2 in this case, from a dendritic cell or an antigen-presenting cell, this cell is going to put it on this MHC2 molecule, and when it shows the world and says, hey, immune system, take a look what I have, now the immune system says, oh, I can see you're not the bad guy because this antigen is actually on an MHC2, but I see that you're showing me the bad guy, right? So these MHC molecules are super, super, super important. All right, so what happens once these things bond together, right? So the antigen receptors on the surface of helper T cells bind to these antigen-presenting cells, signals are exchanged, and whammo, the helper T cell gets activated and proliferates and forms a whole bunch of clones of that, and now these cells can go out and activate other cells, right? So once a helper T cell bonds to an antigen-presenting cell, so long as it has the right receptor, we're in business and we're ready to start this war. Here you can see another graphic of that. So you have your antigen-presenting cell. So he took in, this is phagocytosis, right? He digested this bad guy here, this pathogen, and then these antigens, he expresses, he shows, he presents it to the body, and he does that on a class 2 MHC. So now this helper T cell, who's not yet activated, if he's lucky enough to have the exact right receptor for that antigen, what he does is he touches it and he says, oh boy, is this a bad guy? Well, no, because he also has this accessory protein. And what that does is that touches the MHC and it says, oh, I'm being told that this is the bad guy. Whammo. So there's signals in here, okay, cytokines or cytokines and things like that. And we'll talk a little bit, or I'll show you interleukin 1 and 2 in just a second. But what happens here is this helper T is now activated, and now, wham, we're going to go ahead and elicit an immune response. We're going to get the humoral response going, which has a couple little things that had to happen first, and then the cell-mediated response is also going to get going, where we make these cytotoxic T cells. Really, really complex and cool interactions about to happen.
Now let's look at how helper T cells aid cell-mediated immunity and humoral immunity. Imagine that bacteria have infected the body. Antigen molecules on their surfaces identify them as foreign. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface, along with a self-protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. The antigen-presenting cell secretes interleukin 1. Both interleukin 1 and contact with the antigen-presenting cells activate specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen.
So cytotoxic T cells are a response to infected cells. These are the effector cells in a cell-mediated immune response. So if you remember last time we talked about immunological memory, okay, you're also going to make memory T cells, but these cytotoxic T cells are the ones that are out there actually working for us right away. They recognize fragments of foreign proteins produced by infected cells and possess an accessory protein that bonds to class 1 MHC molecules. And these activated cytotoxic T cells secrete proteins that disrupt membranes of target cells and they trigger apoptosis. So here you can see this is your body cell, right? Okay, it might not look as handsome as this guy or as dangerous, but point is, this is your cell and these are your cells. So this is a really cool immune response because what this guy is actually going to do is destroy your own cells, but he only does that to cells that are infected with the pathogen. Okay, and he can also do that, which is awesome, against cancer cells. So cancer cells often morph into something recognizable by your immune system, and that's why a lot of us, we might have had cancerous cells inside of us, but the disease never took hold because our immune system took care of it for us. Really, really cool. So here you can see another graphic of that. So again, the cytotoxic T cell is going to bond, okay, to the class 1, in this case, MHC. All right, so this is your cell, your body cell, and it got infected, and it's showing everybody, hey, just so you know, everybody, I am infected right now, and here's the guy who's infected me. It's showing its antigen. And the cytotoxic T cell, who's been activated, thanks helper T, to know this guy, he now touches the MHC1 and says, okay, I can tell what's going on here. You're showing me that you're a cell and that you're infected. And then he releases these chemicals, right? Perforin is going to form little pores in the membrane. These granzymes can get in there and start digesting the cell. Leads to like blebbing and such, and kills the cell. So here's a little video. See if this makes it any clearer for you.
Now let's look in more detail at cell-mediated immunity. Cytotoxic T cells are the main warriors of cell-mediated immunity, which attacks body cells that have been altered by infection or cancer, or transplanted cells that are foreign to the body. An infected, cancerous, or transplanted cell displays foreign antigens on its surface. T cell receptors on specific cytotoxic T cells recognize and bind to specific antigens on cells. Cytotoxic T cells respond to this contact and to interleukin-2 sent out by helper T cells by producing the protein perforin. Perforin molecules are released from the cytotoxic T cell and attack the membrane of the infected cell, punching holes in it, which allows water and solutes to enter. The cell bursts and is killed. In this way, cytotoxic T cells deprive pathogens of their host cells so they cannot reproduce. Tumor cells are also removed from the body by cytotoxic T cells. In this way, cell-mediated immunity, carried out by cytotoxic T cells, destroys altered or infected body cells.
So that's pretty simple, right? There is the cytotoxic T cell formation. That is the cell-mediated immune response that you're specifically fighting against a pathogen, right? So it came in, an antigen presenting cell recognized it, ate the virus or the bacterium or whatever, and it presented it. A helper T cell got activated, and in this case, he goes to T cells, and he's like, hey, here's the bad guy, everybody learn him, now go get him. And when the T cells, okay, the effector cells are called cytotoxic T cells, when they see the bad guys, they know that that cell is infected, okay, because the bad guys are being presented on MHC1s, and they kill it, right? So that's the cell-mediated immune response of the adaptive immune system or the acquired immune system. So what's going to happen next is, let's talk about the other way, the B cells, right? The humoral response. So now we're talking about your fluids of your body and antibodies being produced. Here we go. So B cells and antibodies are a response to extracellular pathogens. So the humoral response is characterized by the secretion of antibodies by B cells. So we've already covered over here, right? Now we're going to talk about what happens here. And it is a little bit different. So activation of the humoral immune response involves B cells and helper T, as well as proteins on the surface of pathogens. Okay, in response to cytokines from helper T and an antigen, a B cell proliferates and differentiates into memory cells, of course, and antibody-secreting plasma cells. So the effector cells this time, instead of a cytotoxic T cell, is what we call a plasma cell. Now, why is this one a little bit different than before? These B cells are floating around, okay, in your body fluids, and if they happen to run into a pathogen, a little nasty, that matches their receptor, ooh, now they're thinking, aha, I think I know that there's a bad guy around. For example, look at this B cell right here. He can't bond to this guy. It doesn't fit his receptor. This one doesn't, the antigen doesn't fit the antigen receptor. Okay, and remember these are like little Ys. They look like antibodies, except they have a transmembrane region, right? We talked about that last time. But down here, ooh, it does fit. Now though, just because you found an antigen which matches a receptor, that doesn't mean that this guy right here knows everything. He has to be told, you're right, that is the bad guy, let's get him. So what happens is, now that he's bonded, okay, a bad guy, he has to wait for our immune system all-star, in my opinion, the helper T cell, right? The T helper comes down and says, hey, you did it, you found the bad guy, you're now activated. And now this guy can then differentiate or divide into plasma cells, memory cells, right? And you're going to proliferate a bunch of these things. Now, memory cells, of course, are going to stay around to provide immunological memory, and that's going to be around for hopefully a long time. But the plasma cells are your effector cells, and what they're going to do is they're going to create a whole bunch of antibodies, and you'll see the various ways that antibodies can help us fight disease in just a second. So here's another diagram showing the same thing. So again, it starts with phagocytosis. You have a macrophage or something like that. You have this antigen presenting cell taking in the bad guy, right? Presenting the antigen on an MHC2. The helper T cell then says, oh, I see what you're doing here. And it connects, hopefully, to the correct antigen here. And it also says, I see you're presenting that to me. This helper T cell is now activated. And what he can do is, he can now go around and tell other molecules. So this B cell already learned about this bad guy, but he doesn't necessarily know he's a bad guy yet. So what happens is this helper T cell kind of reinforces, or he congratulates him, right, via these molecules here, and he says, yeah, go ahead, right, you got it. Now go forth and differentiate your cells into, yes, memory cells, because we need to remember who the bad guy is, but also, very important, the plasma cells. And you're going to secrete a whole bunch of antibodies against this exact germ. All right, so let's take a closer look at these antibodies. So they don't kill pathogens, right? They mark the pathogens for destruction, or they goof them up. Let's see here. So plasma cells are producing tons of these Y-shaped proteins, and remember from last time, they have a constant region here, and they also have a variable region, okay? And what's going to happen is, here's a bad guy, here's a pathogen, but it doesn't have to necessarily be a pathogen. As you can see, they also bond sometimes to toxins and prevent the toxins, which have antigens, from entering your cells. Okay, so antibodies are really, really versatile, and they're really awesome. So in neutralization, antibodies bind to the viral surface, and they prevent infection. Think about it. This virus wants to land on your cell, right, on your membrane. And what he does then is, he acts kind of like a hypodermic needle for your skin, right? He injects its genome, whether it's DNA or RNA, he injects that into your cell. Okay, so if he has the ability to land on your cell, he's going to do that, and he's going to inject the dangerous genome inside of your cell. Well, look at this. When it's surrounded by antibodies, he can't even land on your cell. You neutralize the threat. Pretty neat. You can also have opsonization happening. Now, again, antibodies themselves don't kill the bad guys, right? But what they do is they can mark it. Okay, they create a target for macrophages or neutrophils, triggering phagocytosis, right? I like to think of this as like a siren or something like that on top of the bad guy now. It's like the antibodies are screaming, here I am, come and eat me. Pretty cool, right? So you'll eat and digest it, all thanks to those antibodies being bound to it, right? And that's called opsonization. Now, last time I told you how cool the complement system is, right? with all those cascading effects that happen? Well, antigen antibody complexes might bind to a complement protein, which triggers a cascade of complement protein activation, right? And basically what's going to happen here, they're going to create a pore, all right, similar to what we talked about before, only in this case, here's the bad guy, here's the foreign cell, right? Here's the antigen, okay? Basically, what's going to happen is at the antigen, you're going to bond some antibodies. All right, we already get that. But now, thanks plasma cells for making these antibodies for us. But now you have these complement proteins that might come and connect to the antibodies, right? To the other end of the antibody, the constant region. And what's going to happen is they can release their proteins and start to attack the cell. And as you can see here, these proteins can form a pore, and then, oh boy, you're going to have an influx of water and ions. And as you know, based on when we talked about our tonicity, that could be very dangerous and likely just kill this bad guy, kill the pathogen. And that's about it for antibodies for our class. But as you guys go into college and you take some immunity or immunology classes, you're going to learn a whole lot more about antibody function, right? And B cells can express 5 different forms or classes of immunoglobulins or antibodies with similar antigen binding specificity, but different heavy chain regions. So as you can see, maybe all of these antibodies here have the exact same variable region. They will all bond to the same exact antigen. However, look at their other regions, right? They're all different from each other. They can be very different. So basically, and you can see here what they're used for. Okay, and like I said, I'm not going to bother you guys about this, but it's really important, and you'll learn all about them someday. Here's some more about it. So for example, immunoglobulin A, this is found in your mucus, saliva, tears, breast milk, protects against pathogens. Great. Well, immunoglobulin D, part of the B cell receptor, activates basophils and mast cells, right? Look at this one, IgE, protects against parasitic worms, responsible for allergic reactions, right? So really cool stuff here that you can read about. And antibodies are very, very cool and complex. So let's review what we've learned. We have acquired immunity or adaptive immunity. Okay, and if you remember last time we talked about the first or the second response, we're talking about the first response today, the primary immune response. And what happens is, okay, you got infected. There's a bad guy who came in here and infected our body. Now, what's going to happen? Well, we'll talk about this in a second, but there are B cells floating around that maybe have already learned the bad guy, but they didn't necessarily know it was the bad guy. So we started off talking about cell-mediated immune response. So the antigen-presenting cell, okay, or he's going to do phagocytosis, he's a macrophage, and what he's going to do is he's going to express the bad guy's antigen. And what that can do is that can activate a helper T cell. This helper T cell, okay, who is called in, there's a bunch of cell signaling going on here, little molecules floating around. But once this helper T cell is activated, he looks for a cytotoxic T cell that has the correct receptor, okay, for whatever the antigen was that was expressed. And now that the cytotoxic T cells get activated, oh boy, they're going to make, of course, memory cells, right, because you have to have a memory. But they're going to make active cytotoxic T cells, right? And what these guys are going to do is they're going to defend against intracellular pathogens and even cancer. So what these guys do is they travel around, they look for that specific antigen, right? And anybody who has it, they kill them, even though it's your cells. Pretty crazy. Now, what else can happen here? That's the cell-mediated response. What about the response in your fluids, right? The humoral response. Well, that's all about antibodies. And what happens here is this B cell might have already seen this antigen, but he's not ready to go until our all-star of the immune system, the helper T, tells him, okay? Now, what's going to happen here is the helper T says, hey, B cell, I just learned from this antigen-presenting cell that that was a bad guy, right? I'm now activated, and now that he already knows the bad guy, whammo, the chemicals being released by the helper T cells are telling the B cell, go to work, right? And what's going to happen here? Well, you have memory B cells, of course, that travel around all the time, and then you have the plasma cells. The effector cells, in this case, make a whole bunch of antibodies, and you saw how they work. And the beauty of this, not only is the helper T cell doing all of this, it's also making more of itself, right? It's proliferating like crazy, and it's also making memory helper T cells. All of this memory that's existing now, oh baby, the next time we get sick or infected from the exact same antigen, remember the graph we talked about last time? Oh boy, you're going to fight this disease now very, very, very quickly might not even show symptoms. Maybe last time you were very sick, and this time you don't even know, because your immune system has now been it's sensitized, right, to this bad guy, and now you're ready to fight, which is really cool. So that's the typical immune response, right? There's a lot, and this is a broad overview, right? There's a lot more complexity than that, but hopefully this helped you out a little bit, and I hope you can see how important helper T cells are. That's why the disease that a lot of you have learned about already, HIV, which causes the disease AIDS, right? That's very dangerous because your helper T cells are in play, right? So if HIV is affecting your helper T cells, whoa, your whole immune response is going to be messed up. You're going to be very immunocompromised if you're dealing with that disease. Okay, let's go a bit further. Okay, so active and passive immunization. All right, active immunity develops naturally when memory cells form clones in response to infection. That's basically what we've been talking about today, right? It can also develop following immunization, also called vaccination, right? In immunization, a non-pathogenic form of a microbe or part of a microbe elicits an immune response to an immunological memory. Okay, so this is amazing. Now, I could get on my soapbox right now and talk for 3 hours about how important vaccines are. And as your biology teacher, I got to tell you, when you hear people talk about vaccines and the dangers of vaccines, or you know, being hesitant to have their kids vaccinated or their loved ones or things like that, you need to say, hey, we live in a culture right now where people are getting their information from social media and from these blowhards, and there's a lot of people out there that are just sensationalists talking about this stuff. No, get your information from scientists, from people who have spent their entire lives researching these diseases, developing these vaccines, and having these vaccines be tested, right, methodically via the scientific method. Vaccinations are so important, and it's why probably a whole lot of people watching this video are here today, is because of vaccinations, right? So we're recording this right now during the coronavirus pandemic, right? And what's going on? Well, we're all hoping that they make a vaccine soon, right? There's a lot of people dying of this virus right now, and we're hoping that, hey, maybe we'll get a vaccine against it. And what they're going to do is then they're going to take a form of the virus that's not, you know, pathogenic anymore, or they're basically a part of the virus, if you want to think of it that way. And we're going to get that introduced into our immune system, so we recognize the virus. We'll start to fight the bad guy, even though the real bad guy isn't even inside of us. So now when the real bad guy shows up, we've all been inoculated already, right? We've been immunized, right, or vaccinated, so we know how to fight them already. So our secondary response is going to be so fast. Hopefully we destroy that guy before we even have to worry about it inside of our body. So please, if you hear of anybody talking about like vaccinations and things like that, and especially like dangers of vaccination, they used to talk about autism with vaccination and things like that. No basis for truth in there. Matter of fact, the scientist that posted that, he got in huge trouble. He lost his, you know, license and all this stuff. There's no proof of anything like that. Now, could bad things happen? Yeah, very, very small chance that sure, you'll be that person out there where a vaccine, you know, it just worked in a way that it didn't go as well. Well, yeah, maybe you're a 1 in a million shot of that happening, but think of all the other people that did get benefited, right? And that herd immunity that happens to eradicate our planet potentially of these diseases, hugely important. I know teaching a couple of years ago, I had a student whose parents were very upset because they didn't want to vaccinate their kids as much as what doctors said. In other words, why give them all these shots at once? Let's spread them out further. Fair point. I mean, as a father, I worry about my kids all the time. But again, scientists have tested these things. And that's not to say don't question people, right? You should always question. That's what makes this country great. That's what makes being a human being great, right? We're very inquisitive creatures, right? So we're trying to figure out what's best. But point is, no, they've tested this, and they're telling us here's the current best method for protecting you and our whole population against these diseases. Vaccination is very, very important. So what I'm going to do is I'm just going to share a little YouTube video with you right here. That's a cute little video that shows how vaccinations came about from cowpox. Pretty cool.
This is the smallpox virus. It infected the pharaohs, caused a terrible impact on the Greek and Roman empires, and decimated Native American populations. Nothing seemed to stop this lethal plague until the end of the 18th century. At that time, smallpox was a major global endemic disease. Only in Europe, it killed around 400,000 people per year. It didn't differentiate between rich and poor. Even 5 kings were killed. But for some reason, milkmaids and shepherdesses seemed to be immune to the disease. In fact, they were renowned for their beauty because their skins were free of the scars smallpox caused. They were attractive and resistant. But why? That's what the rural doctor Edward Jenner was trying to understand. He knew that milkmaids got small blisters on their hands when they milked cows suffering from cowpox, a mild disease that cattle suffered from, which caused similar lesions to smallpox. And this phenomenon made Jenner think that maybe cowpox and smallpox were related, and that infection with the milder disease would be enough to protect people against the lethal one. As a man of science, he decided to prove it. And to do this, he needed 3 things: cowpox that he extracted from a milkmaid, fresh smallpox from a patient, and a human guinea pig. He chose his gardener's son, an 8-year-old boy called James Phipps. He infected the child with cowpox, waited a couple of months, and injected him with smallpox. And James didn't become ill. He repeated the experiment with other patients with the same results. Jenner was right, even though he didn't fully understand how his remedy worked. The most remarkable thing was that his discoveries were made way before Jacob Henle proposed in 1840 that infectious diseases were caused by germs, a theory demonstrated by Robert Koch. Decades later, Louis Pasteur, inspired by Jenner's experiments, showed that attenuated microbes could be used to prevent diseases, and he proposed the terminus vaccine in his honor. Almost 200 years after Jenner's experiments, and thanks to global vaccination campaigns, in 1980, the World Health Organization announced one of humanity's biggest achievements: the complete eradication of smallpox, the most deadly infectious disease in history.
So how about that? Edward Jenner thinking like, oh, the cowpox ladies who are apparently known for their beauty, yet they still got cowpox? I don't know about that story. But anyway, he said, hey, if they're getting cowpox, which looks just like smallpox, let me go ahead and scratch a little pustule on them, right? Get some cowpox. And then, oh, who can I infect with this disease? Well, let's pick the gardener's son, right? An 8-year-old kid. That's crazy. But what a parent saying, yeah, sure, random doctor, go ahead and stab my kid with some disease. So this kid then got cowpox, right? But his immune system learned, and because he's making now immune cells that are fighting a disease with an antigen that's super similar to smallpox, boom, there we go, right? Now we developed immunity. And years later, now that they understood how this was happening, sure, they developed vaccines. You didn't have to take the pustules anymore and take like oozing stuff from people and inoculate them with it. Now you could just take a needle with an attenuated form or things like that. Pretty awesome. So vaccines, super, super, super duper important. So passive immunity provides immediate short-term protection. Okay, and it's conferred naturally when IgG crosses the placenta from your mom to the fetus, or when IgA passes from the mom to the infant via breast milk. Right? Very, very neat that before you're even born, you're getting antibodies, and shortly after you're born, you're still getting antibodies, right? So super important here, because obviously when you're the hardest time of your life, the most dangerous part of you was when you were a fetus, right? When you were inside your mom. A lot of things can go wrong there, so we're lucky to be here, right? And part of that is because of your mom's immune system taking care of you. Another thing that can happen then is after you're born, whoa, you're in this crazy world with all these pathogens all over the place. Well, thanks to breast milk, your mom is still giving you antibodies. Okay, and of course, there's other types of milk now available to kids. So maybe you're not getting that, but there's research that shows lots of different reasons why, yeah, breast milk is very good. Okay, and it keeps us safe. You can also get artificially conferred antibodies, right? So you can inject them into a non-immune person. Right? Like I said, we're actually, I'm recording this during the coronavirus pandemic, okay, of 2019, 2020. And what's going on is a lot of people that already beat the disease, right? They already have antibodies circulating throughout their blood. So what a lot of scientists right now are researching is, hey, can't we just take that person's blood? Okay, and there's really cool ways I'll talk about in a second to get the antibodies out and things like that. But can't we take that person's blood and give it to people who are very sick, and maybe those antibodies will help out, right? It's like a passive immunity. You can't learn the virus because it's killing you. You need a boost, so sure, give them IVIG, right? Intravenous immunoglobulins or antibodies. Pretty neat. Scientists are currently doing a great job of using antibodies as tools, right? So the antibody specificity and antigen-antibody binding have been harnessed in research, diagnoses, and therapy. Polyclonal antibodies, which are produced after exposure to a microbial antigen, are products of many different clones of plasma cells, each specific for a different epitope, right? So we can have all different types of antibodies. We also have monoclonal antibodies, prepared from a single clone of B cells grown in culture. And these can be trained to just attack, right, the one specific disease. And oh boy, the textbook I use to teach this course is getting pretty old now, to be quite honest with you. But yes, this monoclonal antibodies being used to fight cancer is just awesome. It's pretty miraculous what we're doing right now. So neat stuff ahead, and it's using your immune system and using even sometimes pathogens, right, which we make safer now to deliver certain things. We're using your own immune system to fight diseases by training it in different ways. It's just amazing what's happening right now. And let's talk about immune rejection. I have a little story for you here. So cells transferred from one person to another can be attacked by immune defenses, of course. This complicates blood transfusion or the transplant of tissues or organs. So these people right here, I know both of them very well. This is my sister-in-law, Amy, my wife's sister, and that's their cousin, Brian. Okay, and I guess my cousin now, right? Brian. So what happened was Brian was dying. Brian needed a kidney very badly. And this happened many, many, many years ago. Okay? And what was going on is, well, at the time, you had to wait for a donor, right? You have this thing, you know, your MHC, major histocompatibility complex, things like that. Well, humans have HLA, a human leukocyte antigen. Okay, and what you're trying to do is you're trying to match up a donor. You're trying to find somebody who's very similar to you. So your immune system says, right? You have all those little feelers out there and receptors. It says, I think you're okay. I think you might be me. So it doesn't attack. But anybody else's organ that's in there, if you're not a super close relative or got very lucky with a match, you're going to attack it. You're going to kill it. So here you are trying to save somebody's life by giving them an organ, and if it's not a match, they'll die. Well, they didn't know this at the time, but they were actually part of a study being done at Johns Hopkins University. And basically, the New England Journal of Medicine has this published article, and they are part of this study where what they did was, instead of waiting for a donor, because Brian was going to die, they destroyed his immune system, basically. Okay, and that's a little dramatic way of putting it, but they did this thing called plasmapheresis, where they take out his blood and destroy the immune components, put it back in, but now he could get a non-matching kidney. So Amy was able to give one of her kidneys. What a selfless person that is, right? That's nuts. And she's doing well today with her one kidney. But she gave her extra kidney to Brian, who's still doing very well today, all because his immune system didn't attack it, because they damaged his immune system. And eventually his own body kind of takes over that kidney and makes it his own. And he's doing quite well right now. Pretty awesome, right? And this is now pretty much well known, and this is how a lot of organ transplants go. Sure, it's better to have an exact match if you can, okay, but yeah, a lot of times you just do this and you just knock down the immune system a bit. Had to be very careful. We weren't allowed to go visit, you know, Brian after the surgery because his immune system was low, so any germs we brought in could be very dangerous for him, whereas we have strong immune systems, but he was immunocompromised, right? Pretty neat stuff. So just to repeat that a bit, you have MHC molecules are different among genetically non-identical individuals. So if you're lucky enough to have an identical twin, there you go. You got some organs ready to roll if you need them. Differences in MHC molecules stimulate rejection of these grafts or organ transplants. And chances of successful transplantation increase if donor and recipient MHC tissue types are well matched. But you can give immunosuppressive drugs. And like I said, there's other methods too to get rid of immune system parts. And lymphocytes and bone marrow transplants may cause the donor tissue to reject the recipient. So, could be scary. And like I said, MHC, we also have this HLA is a short version in human leukocyte antigen. Okay, and MHC is found in vertebrates, but HLA is only found in humans. So different ways to tissue type. So let's wrap up today talking about something we have already covered in genetics, right? Blood groups. And you remember that there are antigens on your blood cells, right? This is old school here. So remember, if you're type A blood, what does that actually mean? That means that you had this little enzyme that connected an A antigen onto your blood cells. So you have this little spike, if you will, on your blood cells, right? Little A's. If you're type B, sure, you have type B spikes all over the place, right? Little antigens. And if you're type AB, which was codominant, right? This is a multiple allelic, could be codominant, okay, phenotype here with AB blood, you have both, right? You have A and B little spikes on your blood cells. And if you're type O, right? Recessive, homozygous recessive, you don't have any of these spikes. You have no antigens. And that's why we said, well, if you're type AB blood, hey, you're the universal recipient. Because if somebody gives you type A blood, your immune cells say, yeah, I know you. You're A, you're good. Right? If somebody gives you type B and you're AB, yep, I know you, you're good. If somebody gives you type O, you're always okay, right? So AB is the universal recipient, whereas type O is the universal donor because everybody can get type O. Unfortunately for type O, though, you have to get type O because any of those antigens your immune cells will attack. And it does happen where sometimes you create, okay, an immune response through a bad transfusion or through somebody doing this, not matching up the blood group correctly, right? So transfusion with incompatible blood leads to destruction of the transfused cells, okay? So if you have recipient-donor combinations going on, they can be fatal or they can be safe, and hopefully people protected it, okay, or double-checked. You might be asking yourself about the positive and negative again. Remember, that was that rhesus factor, the Rh factor. And we did talk about that a little bit too, because that's kind of crazy. So what if a mother was Rh negative, but the father, okay, is Rh positive or can donate Rh positive? Well, if that baby is born Rh positive, that's okay, right? Because the mom isn't really attacking that baby going on because she doesn't know anything about this positive Rh, you know, little antigen hanging off of the blood. However, after birth, sometimes the mom does get sensitized. Sometimes the mom's immune system realizes, wait, what is this Rh positive blood? But the baby's already gone, right? So no big deal. But what about the second time the mom gets pregnant? If she has immunological memory built up against Rh, or she was sensitized to Rh positive, well, now you have to be very careful, right? And that's why there's so many appointments that you have to go through and things like that when you're going to have a baby, so everybody can keep track and make sure that you're not going to attack the fetus that's growing inside of you. Pretty cool. So that was a little bit about the adaptive or acquired immune system. And if you're my students now, we probably watch some bioflicks here and get into more visual representation of what happened. But hopefully this made sense today. Hopefully you now understand that there's two main legs to acquired immunity or adaptive immunity. You have the cell-mediated response where the cytotoxic T cells go out and kill infected cells or sometimes even cancer. And then you have the humoral response where we make antibodies, right? And those antibodies, I showed you a bunch of different ways that they can actually help us out, right? We talked a little bit about vaccinations and why they're so important, right? And you don't have to deal with these sicknesses if you could just get vaccinated against it, right? When I was a little kid, I got chicken pox. You guys probably watching this video don't have to worry about chicken pox, right? When I was young, this herpes simplex goes around, chicken pox it's called, and I would get it. And sometimes parents would even have parties where they get all the kids together and they all play and stuff like that. So we all got chicken pox at the same time, usually over the summer. So we'd be scratching and itching with all these little red bumps, but at least we didn't have to miss school and things like that. So our parents would have chicken pox parties over the summer. You guys got a vaccine, so you don't even have to worry about it. Pretty neat, right? The vaccines are amazing. And now, do I have to worry about chickenpox ever again, or do you have to worry? Well, it can come back as an adult. The virus lays latent sometimes in your nerve cells and can come back as something called shingles. But man, vaccines are the way to go. They're so important, right? And then we also talked about tissue transplants and blood, you know, matches and things like that. So very important. So I hope this video was helpful to you. And the immune system, if you can't tell, is my favorite system. It's amazing. And I hope you enjoyed it. Okay, take care.